I'm sure you've all heard of the cervical cancer vaccine which rolled out in 2009. And I'm sure you all know what a major milestone it was in the fight against cancer and in the protection of the women of our nation. What you may not know is that we may not actually see the benefits of this vaccine for up to 30 years due to the time it takes cervical cancer to develop. What you may not know is that the virus which causes cervical cancer, called HPV, also causes 6 other types of cancers, affecting over 700,000 women and men worldwide in 2012. What you may not know is that there is no specific treatment for HPV-caused cancer. This means that those 700,000 women and men will face what we, in the Macmillan Research Group, call the medieval torture forms of treatment, including slicing, cutting, burning, and poisoning. These torture treatments describe treatments with adverse side effects, such as radiotherapy, chemotherapy, and invasive surgeries, such as the surgical removal of the entire female reproductive organs. This is where our research comes in. But how do you develop a treatment which will only kill HPV-infected cells and leave all other cells unharmed? How do we improve upon current treatments and reduce side effects? Well, the answer lies in our approach to a treatment, and the concept behind it isn't too difficult to understand. The easiest way for me to explain our treatment is to imagine that all cells in your body have 2 motors which keep them running and living and dividing. Now, when HPV infects a cell, it'll effectively destroy one of these motors. This cell can keep living, though, because it still has 1 motor left. What our treatment does is we've figured out a way to turn off this second motor. Therefore, the cancerous cell will die. This treatment leaves all other cells unharmed because we are only turning off the second motor and they still have the first motor to keep them running. This effect is called the synthetic lethality effect, and this is because we are inducing a specific lethal effect in only HPV-infected cells and leaving all other cells unharmed. My research asked not only if this therapy could work, but how it works and why it works. I want to know what's so special about these 2 motors, and is there other ways we could turn them off? How could we use this knowledge to our advantage? By answering these questions, I aim to provide a shred of hope for those 700,000 people currently suffering with the disease and all future implicated generations. Thank you.